So for our last Grand Round seminar for the CRDEB of the WVCTSI, we have, I think it's going to be an exciting guest. It's Dr. Charity Moore, who's visiting us from the University of Pittsburgh, where she is a professor of medicine in the Division of General Internal Medicine, Department of Medicine, School of Medicine. Lots of medicine there. But she's also currently the core faculty member for the Center of Research on Healthcare, core faculty member at the Institute for Clinical Research Education, the ICRE, the director of the Clinical Trials Research Track, Masters of Science program in Clinical Research, an affiliated faculty member in the section of Healthcare Disparities and International something, I cut it off, and the director of academic programs in the ICRE at the University of Pittsburgh.
That's the most important one.
There we go. And she currently holds membership with the American Statistical Association and the Society for Clinical Trials, and is maintaining very active activity at all service levels, both at the university all the way up through a national and, my mind just blank. The interesting part is with all these medical associations, she did get her PhD in biostatistics in 2000 at the University of South Carolina. So I think she's going to have a very interesting talk for us. Also, a bit more of her history. She has 11 grants and has been involved with approximately 20 grants over her time. And she has over 50 presentations, 30 of which are invited and are very biostatistical in nature, dealing with issues of secondary data, clinical trials, and exploratory studies. She has over 170 publications, including almost 100 first and co-authored publications in a wide range of topics. So I will just skip over that wide range, but she's got a very extensive teaching portfolio in biostatistics and in clinical trials. And she's been involved with over 50 master's, doctoral, and PhD students at a variety of universities, and has an extensive mentoring portfolio. So I think she's going to bring to us a wealth of information and a wealth of insight, and I'm very excited about this. So I want you to also join me in welcoming Dr. Charity Moore and thanking her for coming from Pittsburgh to talk to us about developing pilot studies in clinical and translational science. I just want to note that if you have questions, throw up your hand, and for our remote sites, just kind of wave vigorously, and hopefully she will catch, you'll catch her eye in the monitor here. Thank you.
Okay, so I just advanced the slides by clicking, is that right? Yeah. So thank you all for having me. I feel tired now after hearing of all the things that I'm responsible for. But this talk, I am a biostatistician by training. So my master's and my PhD are both in biostatistics from the University of South Carolina. This talk came out of some very applied work that I was doing when I was at UNC. So about 8-9 years ago, I was working on collaborating with an investigator who was wanted to develop a pilot study. And I have to be honest with you, the first time that we submitted this, I did it wrong. Okay, so you have to learn from your experiences. So when I worked with him on his power calculations, I did it wrong. And it was actually a really nice biostatistician who called me and said, this is a pilot study. So what that prompted me to do was do a very thorough investigation, literature search, and to find out how I could not only improve his project, but also improve my knowledge on the topic. So what I'm going to talk to you about today is really to help you, whether you're a biostatistician and you're collaborating with people, or you're an investigator, or if you're a student or planning on collaborating, how you help people design these and plan for these. So I think there's one formula in the entire set of slides. This is meant to be very applicable to those of you who are collaborating and trying to develop these kinds of studies. So there's not any formulas, there's not going to be any derivations. Perfect for a Friday, right? You don't have to engage too much. So one of my roles at the university with the CTSA, ours is called CTSI, Clinical and Translational Science Institute, was to review, when you, even I think at your institution, there's pilot funding with the pilot core, and you can submit an application to get the pilot funding. And so I agreed to serve as a reviewer for one of the pilots that went around. And so the pilot study that I modified it a bit, but it went like this. So the aim was to determine the feasibility of testing an intervention in older women with rheumatoid arthritis. So it's very generic. I didn't want to give you too much information, but we're just looking to test the feasibility. So let's see how this goes. So they were planning on doing a randomized trial, single blind compared to usual care. 20 women are going to be randomized. They were going to monitor them at baseline in 16 weeks, and they were going to get 12 different outcomes. How do you feel about that? 12 probably is a lower bound, right? So when I first read it, I thought, okay, there's a lot of information that's being collected on these 20 women if this is just a pilot study. And so the next thing, and the reason that I say don't do this is my advice would be not to do this. So this was the analysis plan. The statistical procedures will be used as appropriate, as needed, as appropriate. And the sample size justification, there was no sample size analysis provided due to the nature of the pilot nature of the study. And so this happens a lot. And particularly for anybody that's planning a pilot study, they don't think that the justification of sample size is needed because it's just a small study. And there's not an analysis plan that's needed because it's a pilot study. So then I would ask you, would you let your mother, spouse, or you yourself, would you enroll in this trial? Not sure what that looks like, but would you? No. Because it's clear that the investigators don't even know what to do with the data that they already have. So I gave it a very bad write-up. I'm not sure why it went. I gave it a bad write-up, and ultimately, I don't know if they got funded or not. But I still see this happening a lot. So it's just to give you an example. So what we'll do now is talk about what might be a better strategy. So let's see if we can get this to work. Did it just go to sleep?
I'm not too sure. When I came there.
Okay. Okay, so why should you need to know more about designing good pilot studies? Well, one of them is they're highly prevalent in health sciences research. Everybody's got to start somewhere, whether it's because you've got very little funding, but you basically have to establish that you're an investigator and you can do these things. So we see a lot of them. There are a lot of funding opportunities, little pockets of money here and there that you can get to do pilot funding, to do pilot studies. And you need this to establish yourself to get the next study. So they're very important. I can make sure that you have access to the slides, but just to give you a snapshot, there's some really good articles. The first one, of course, is mine. But I worked with 3 other statisticians to write an article about pilot studies. And we wanted to write it not for the statistical audience, but for investigators to use when they're planning their studies. So this was published in Clinical and Translational Science. It was also because we were all part of CTSAs, and so being funded, our efforts were being funded by the CTSAs as well. And there are a couple others. One is by Bruce Dobkin, who is mainly doing more rehab and motor intervention, so it's very applicable to those areas. And then another one in critical care. So I can make sure that you get a copy of these if you need them. Here are the other reasons. So these are a lot bigger reasons, and telling you where NIH stands on this. So the first one, these are all funding, NIH funding mechanisms. One's an R01 to develop a pilot study to look at novel, low-cost methods for clinical trials, because clinical trials get a bad rep for being expensive and too many resources, and so they wanted to fund that. There's an R34 where you're planning the infrastructure for a trial, but they're calling it pilot studies. There's an R21 from NIAMS where they're planning pilot studies. So there's a lot of money, and the key is that if you're going to do a pilot study, you need to do it right and plan it correctly. There's also R34s, which fund pilot studies, and again, the R21s. But you have to look at, this is interesting, so one of them, for the R34s, it says, for example, and this would be in the RFA, test the feasibility of an outcome or an intervention in the field. So the focus here is feasibility. But then when you go to an R21, and they're talking about pilot, it's developing biomarkers and imaging. So you, depending on the institute within, or the funder, you kind of have to look at what their definition is of pilot study. So long ago when I started working on this, I just looked up, well, what's the definition? And I really like this one because I think it helps us plan our next steps. So the definition that I found that's easiest is this should be a preparatory investigation. And it's the aspects of your full study, bold italics, that are novel, untested, complex, or innovative. So that means, as an investigator and as the statistician working with the investigator, we have to work to think about what's the next study look like, right? Before we can plan the pilot study. So you have to know what the big study or the full study or the next step looks like. And now you're seeing that a lot more interwoven into the funding announcements, that you have to say where this is going, what's the next step. So in drug trials and in drug development, early stage studies all the way up to phase 3s are fairly well outlined. So you do the preclinical, whether it's the animal work, then you go into phase 1 where you're looking at safety profiles, toxicity, phase 2 with short-term efficacy, and then the go, no-go decisions are made whether you go on to phase 3. What we found in working with junior investigators was that people were doing pilot studies, or they were calling them pilot studies, but it was never quite clear what was having them move on to the next study. And so ultimately, our concern was, because we were working with junior investigators who are trying to launch their careers, that they were ending up in this cycle of not having any productivity. So what would happen is they say, okay, I'm going to do this new project. I'm going to test the research. So I've got a small study. I'm going to test it. It's really a pilot. And then their p-value, for whatever, would be greater than 0.05. And so they'd say, oh, well, let's not do that anymore. We're going to go on to a new research topic. So ultimately what would happen is I've invested 6 months working with somebody on a project. They've invested money and resources and human resources to work on a project. And now at the end of this, we don't have a publication. And we probably won't even have a grant fed off of the pilot study. And so we found this as actually detrimental not only to science, but also to our careers, because there were a lot of pilot studies being done that never were published. So we thought we would do better by giving guidance to people about, if you're going to do a pilot study, design it well in the hopes that all of them should see the light of day in scientific journals. So don't start a pilot study without the intent of actually publishing it, because we all can learn from others' experiences. So here are the reasons you want to do it. One is to design or assess a research protocol. Another one that you're going to see a lot in the funding announcements is just to test the feasibility of something. So is this study even is it feasible? I can give you examples of lots of studies that we've rolled out, and they weren't feasible. And some really good pilot studies would have helped us to determine that. Possibly to establish in the sampling frame. So if you were going to do a survey and you wanted to be able to call people on their home lines, home phones, that's probably not going to work nowadays, right? Because a lot of people are switching over to cell phones. Or if you wanted, if there were a certain number of census tracts that you wanted to, so a good pilot study could establish that that was going to work. Another reason to do a pilot study is if you want to estimate some baseline rates. So for future power calculations, if you wanted to look at rates of a response, maybe let's say in a control group, or maybe the variability in your outcomes. Let's say you're measuring quality of life or some kind of continuous outcome. And I think this is the last one. When I was doing research on pilot studies, one of the things that came more from, I wouldn't say basic science, but much more in the translational work, was this proof of concept. So I consider it more of a mechanistic study. So if you say you're going to deliver this drug and you're going to hit this target, does that target actually move? And so that's a reason to do a pilot study, but it really gets away from feasibility. It's not about can you deliver the intervention, is the intervention shifting its target? And I'll give you some examples. So depending on the question for the pilot study, then we get into, well, what's the study design of the pilot study? Is it a single cohort with a pre-post? Is it a randomized controlled trial? Is it just a cohort study where you measure people at 2 or 3 or 4 time points? Is it a cross-sectional study where you just do a simple survey? Well, it really depends on what your bigger study looks like. Remember, the definition of pilot study said you have to know what your full study is before you can plan the pilot. So if you were going to do a big randomized controlled trial, you really need to do a pilot to make sure that everything works before you spend a lot of money trying to get it to work. Or if you were going to do a survey of people over time, you might want to do just maybe a cross-sectional study to see if that's feasible. So again, it depends on what the larger study looks like, and it also depends on what's being tested in the pilot. So if you were going to test in a pilot clinical trial, sometimes what I've seen happen is people want to do a big clinical trial, and then they'll say, well, can we actually recruit people to get into the trial? So let's say that you did a sample size calculation, the big trial has to have 300 people. And then you just do a survey in a waiting room and say, would you be willing to be in a study that looks at, let's say, Parkinson's disease progression. And people say yes, they'd be willing to be in the study. That's a different question than, would you be willing to be in a study and be randomized to this type of care versus this type of care? So it's a different question to ask people, would you be in a study versus being willing to be randomized? Another slightly different question is, you may be able to get physicians to refer patients to you to come into a study, just a cohort study. But to have physicians send you patients or refer patients to you where your patients will be randomized to do two different types of care is a totally different question. And so you could test in different frameworks through a pilot, different recruitment methods and what was going to work. So ultimately, we have to justify our sample size. So the bad example I gave you, and I've seen this quite a bit, is you don't have to justify sample size if it's a pilot study. Okay? So what keeps us from entering 20 people into our study, 20 women into our study, versus 10, versus 30? So I've worked in some diseases, hemophilia is one of them, which is a rare disease. And it would take us maybe 3 years to get 20 people at one site. Maybe rheumatoid arthritis is more prevalent, it's easier to get 20, but we still have to justify how many people are coming in the study, particularly relative to risk. So this is the only formula. What is this a formula for? Sample size. For what? Power for?
Two sample tests.
Yeah, 2 sample tests comparing 2 groups. So this would be the easiest thing that you could do for a clinical trial, to compare one outcome at a certain time point. And that's the delta, the difference between the 2 groups in the denominator. And then, of course, you've got your alpha level and your power level and then your variance parameter. So this is not the method that we would use to justify sample size in a pilot study. Because if you're testing the feasibility of bringing people in and delivering an intervention, that has nothing to do with efficacy of your intervention. And so this is not the pathway we would take. So I'll give you some examples, but right now I'm more of the abstract. We can talk about, well, it really just depends on how your outcome is defined. So if you said, we really just want to see if we can deliver the intervention. So some interventions are very difficult to deliver. It's hard to get people in and get them to actually maybe even get, let's say, 8 out of 10 sessions delivered, whatever the intervention is. So you could dichotomize. So I've worked with people who say, well, we just want to see if it's feasible. Well, what's the definition of feasible in your study? And so a lot of times that could be dichotomized based on whatever their definition is. Or maybe even if it's safety, particularly if you're dealing more with a drug. So there's different rules of thumb, ROT, rules of thumb. If the purpose of your pilot study is to be able to estimate maybe means for a control group or for just kind of a natural course of a disease, or if you want to get standard deviations, because you need that, of course, for the larger study with your variance. There's a couple different rules of thumb. One of them, Dr. Carter's, I think it was, I'm having a hard time remembering remember in the reference, but one of them references that per parameter you want about 30 people or about 30 independent observations. So if you're going to estimate standard deviation, you want about 30 observations. But if I'm working with my hematologist investigator and she says to me, well, hemophilia, it'll take me 2 years to get 30 people in the study. So that really doesn't work. There was another article that was, when I worked with Dr. Carter that he sent to me that says, really if you want standard deviations, if you get about 12 people into your study, it kind of levels off how much more information that you actually gain, and so you could use about 12. So those are just rules of thumb. If you have more mechanistic outcome, you can actually use the two-sample test that I showed you before, and there's a power calculation that goes into it. But that's very different from having a feasibility outcome. And I have some examples. So then Dr. Stewart, who was my mentor at UNC, and he's on our paper, said, but then you've got all these other things, okay? So if we can, as statisticians, we can crank out a formula, run a couple of things, put some confidence intervals around it and say you'll have this much precision. But ultimately, it comes down to how many hemophilia patients do you have, at this site. So that's a participant availability. How much research burden is it for the patient? How many of you have had an MRI? Yeah, sorry, that's kind of, I guess, private. I have one. We make our patients in our study have 2-hour-long MRIs for one of our trials. And part of the problem now is that patients don't want to do this anymore. So we're getting our primary outcome for our study. And for those of you who have had one, I was in it for 20 minutes. And it was awful. But asking people to go through the motions of 2 hours of procedures is a big deal. And so you really have to think about how many people do you want to push to get, something like that. Or draw blood, or 4 hours of questionnaires. So we really have to think about the burden that we're putting on them. Or for us in Pittsburgh, actually driving into the university, which is not easy. There's the risk that's involved, so if you have something that has high toxicity, blood draw is not too bad. And then budget restrictions. So again, going back to the MRIs, they're very expensive. And I know at Pittsburgh, they've gone from really cheap for research costs to very expensive. And so $25,000 won't get you a lot in terms of images. And it also depends on the stage of research. So if you're an early stage investigator, it's probably not a good idea for you to try to roll out a multi-center clinical trial as a pilot. Okay, so here's what you have to have. Regardless, if you're doing it's just a small study, it's just a pilot, you're filling out the grant application for your internal award, this is what you have to have. Good data management, which is not an Excel file, okay? So data management is not an Excel file. You need to have something like an Access database that's relational, or REDCap is free, and your institution would have access to that. So that's been sponsored by the CTSAs. But you need something that's a relational database that you can have data checks either on fields with minimums and maximums, but the data that are going in there are quality and they cannot be manipulated in a way that could, like with Excel with a simple sort, you can get the wrong answer. And you also need an analysis plan. So you can't just say, we'll use stats as appropriate, as needed. So most of what I would recommend for a pilot really needs to be descriptive. And at best, maybe have some confidence intervals so that you're showing how imprecise your measures are, given that you have a small sample size. And if you are doing hypothesis testing, particularly if you have a mechanistic outcome, then it would be good that if you do the hypothesis testing, that you always disclose that it should be interpreted with caution because you're talking about small sample sizes. And then the last component that you need to make sure that you have, although most of the time in the funding announcements, they're requiring this, what are the next steps? So if we see this, we're going to go on to this. Okay? If we see this response, we'll go on to this study. If we see that this is feasible, this is the next step for us. Okay? And it's got to be clear. For the ones that I've reviewed, if you don't have this component, we don't review it. So it's automatically kicked out. Okay. So if you're going on to plan the larger study, so let's say that you want to go on, I specialize in clinical trials, randomized controlled trials, and people come to me all the time to do sample size calculations. So the things that, and I think all of you are familiar with this, but the things that go into a sample size calculation are, of course, your hypothesis. You think drug A is better than drug B. You have to have the statistical method, whether your outcome or your testing proportions or a continuous outcome. We decide what alpha and beta are going to be. And then this really hard part. What's the clinically meaningful difference between the two groups that you want to be able to detect? Sometimes people will call it an effect size if it's standardized. And then we also have to have some information about either the standard deviation, so that's the variation, or if you're looking at some kind of response rate, how people in the control group respond. So there's lots of different pieces that go into this. So for a pilot study, and I'll show you why, you don't want to take the pilot effect size and plug it into the equation that I showed you before. But it is okay to go ahead and include the estimate of variation as one of your possibilities in your sample size analysis and then the control data as well. So I'll show you why. But here's where this comes from. I was working with junior investigators and I said, well, my mentor said, well, we could run this pilot trial, and then whatever the effect size is, we'll take that and power our larger study. Have you heard that before? So we'll take the effect size, let's say it's 0.5, and that'll be our effect size to plan the larger trial. Okay. So Helena Kramer, I'm not sure, I think she's a statistician, but she's mainly done work in psychiatry, wrote a really nice paper about why that mentality about taking an effect size from a pilot study to make a decision about the larger study and whether it goes on, why that's not a good idea. So this graph, what this shows you is, this is at the bottom of it, so this is the probability that the effect size is less than, the observed effect size is less than 0.5 given the true effect size. So what's along the x-axis is the true effect size. And for those of you who might be familiar with effect sizes, a 0.5 is seen as kind of a moderate effect size. I think 0.2 and 0.3 are considered small, and then 0.8 is considered to be a large effect size. These are relative to standard deviations. Okay. So here's how we interpret this wording. If we see an effect size less than 0.5 in our pilot, we won't go forward with the next study. Okay? If the effect size in our pilot trial is less than 0.5, we won't move forward with our next study. So what you see on the y-axis is the probability of aborting the next study. So it's the probability that you won't go on to the next study. Okay? So true effect size on the bottom, and then not going on to the next study on the y-axis. So what I have highlighted there, and hopefully the arrow is actually underneath the place where the lines converge. So if you have a promising effect size in truth, which we'll never know, but let's say that you really do have a drug or an intervention that has a moderate effect size of 0.5, there's a 50-50 chance you can basically flip a coin based on your decision rule of whether you're going to go on to the next study or not, because of what you've declared as if we see an effect size less than 0.5, we're not doing the next study. And so you're making a decision in that pilot study, regardless of the size of the pilot study, and there's a 50% chance with a truly effective intervention that you won't go on to the next study. And so then what she goes on to show is that if you then take the effect size from what you observe, so let's say that you run your pilot trial, the statistician helps you, does the analysis, and you end up with an effect size of 0.6. Okay, so from your pilot, you have an effect size of 0.6. The true effect size is 0.5. But you use the 0.6 in your power calculations for the next study. So what happens with your next study?
Too small.
It's too small. Because the true effect size is 0.5, although we don't know it. But you took this 0.6 from a pilot study that has imprecision around it, right? There's a confidence interval around that 0.6. And so ultimately what happens is the effect size from your pilot study, if you use that for the future study, your future study is underpowered. And so the red bar says if you really base your, this is a priori. If you really base your power calculations for the larger study on something that's clinically meaningful, not the pilot study effects, then you have the true power that you think that you do. Otherwise, you're always going to be underpowered. So it's not so easy to explain, but the paper is very powerful. I'd be happy to share it with you, but I just try to get people away from that, and we'll do the pilot study, take the effect size from that, and to power the larger. It doesn't work. So the likely things that will happen, one is you won't move forward with an intervention that really should be moved forward, and then if you use that effect size, then your next study would be too small. Okay, so now I'm going to get into some examples. So I tried to pull some examples, one that is a grant application, and then several others where people have actually published pilot studies, and I think that they've been done very well, and they're very effective. And even though they're pilots, still move science forward and still help us as investigators plan and do things. So one of them, this was a K award that I helped with when I was at UNC, and so I changed it a little, but it was very much set up like this. So the aim of the study, this was, we wanted to look at performing a feasibility study of the Moore intervention, so it's kind of a joke that I would be the one that was leading the intervention. So the specific aim, or how we were going to do this, was to conduct a randomized study at elementary schools. So we're going to randomize elementary schools, 4 and 4, 4 in the intervention, 4 in the control, 40 second graders per school. And we're going to look at the feasibility of the intervention to provide data to inform a large multi-site randomized controlled trial, and then we've got several outcomes. So you can imagine if you wanted to do an elementary school intervention, this would be a really good place to see if you can even get into the schools and get them to agree to be randomized, recruit 40 kids per school. And I volunteered to coach basketball for my 8-year-old, he's a boy, and see if you can actually deliver the intervention. So it was 7 8-year-olds in one place is not an easy thing to keep their focus. So you might want to see if you can actually deliver the intervention with second graders. All right, so here are the outcomes. Determine the ability to recruit. So I tried to highlight, what's most important. They're all process outcomes. Can we recruit? Can we deliver the intervention? Training, implementation, data collection, and can you actually measure the outcomes? So it may be that you want these kids to do some type of assessment. Maybe you just want to get their BMI, whatever. But you have to be able to show, if you're going to go for millions and millions of dollars for a larger study, you have to be able to show that you can get into the schools and actually do it. So this really broke it down about what she considered to be feasibility. And then there was a statistical component in terms of the outcomes. So if you're going to run a cluster randomized trial, which this one would be, a lot of times that in terms of those power calculations, one of the components that we need is not just the standard deviation, but we need to know how correlated the kids are within a school. And so one of the things that they added on to this is we'll also get an estimate of the interclass correlation coefficient. Now you would use this in conjunction with what had been published in the literature, but it would at least give you an idea of, because this ICC changes, so depending on the outcome that you're measuring, you can even get different ICCs, and so that would be helpful for planning the next study. This is an example of another study, and it was published in a high-level journal. So this was a local recruitment experience in a study comparing the effectiveness of LGID as low glycemic index diet with a low, I think, carbohydrate healthy eating diet in women with PCOS, which is polycystic ovary syndrome. So, yeah, okay. I see some hints to it. Thank you. So the objective of the trial was to look at the feasibility of the trial in these women. So let's just look and see what they their main outcomes were how many people would actually be eligible based on their eligibility criteria. So you'd have inclusion and exclusion criteria. How many people could they find? that fit what they were looking for. And how many people could actually get into their study. So it's one thing to say, well, there's 100 women, but to get those 100 women into the study is totally different. Eligibility, recruitment. Do they comply with the diet? How many of you have complied with the diet for 6 months? Okay, me neither. So it's that was one of the things that they wanted to see. And then they also wanted to see, could you actually get people in and get them measured? So this is more of an attrition component. So compliance to the intervention, and then also coming in for their assessments. And so this is what they found, which is incredibly helpful. Matt's already seen this, but everybody should have to do this if they're going to do a trial. So over 12 months, there were about 4,000 women across 153 OBGYN clinics. So this is the kind of thing that you would put in a grant application. If you were trying to get this trial funded, you'd say, we've got 4,000 women in these clinics, okay? Because it's a big number. 441 of them were actually almost eligible for the study, okay? So now we're down to 11% of that 4,000, potentially eligible. 19 of those actually met the eligibility criteria. So now we're down to 0.4% of the 4,000. 11 consented, so you've almost lost half of the 19 who are eligible, and essentially 8 of them completed this study. This is what I call the Katie Couric way of presenting results. You get 73% hit rate, but it's really 8 out of, I guess, 441. Just depends on your denominator, right? But ultimately, 8 people completed the study out of really what were 441 people who were potentially eligible, but 19 met the trial criteria. So ultimately, I guess when they planned the next study, their entry criteria are probably too strict. So their conclusion was that a multi-center trial would be needed. Remember, they got this from 153 clinics. They got 8 women. So you'd need a multi-center trial. Overall, their compliance was really good for these 8 women. I don't know how generalizable that is. And they gave themselves kudos for doing a pilot study. They thought it was very informative, and I'm sure their future funder thought it was informative too. And maybe they didn't get funded. Here's another study that was funded by NINDS, which is the Neurological Disease and Stroke out of the NIH. This was the GRASP trial. So their goal, with any clinical trial, you have to have a good acronym. So this was the GRASP trial. Their goal was to assess the feasibility and safety of 2 insulin infusion protocol targets in patients with acute ischemic stroke. So that's where the GRASP comes from. So this study, and I've read it a lot, and actually they have the bigger trial was funded as well. What they're doing is saying, these people are coming in with acute ischemic stroke. They're going to get insulin infusion, but they're trying to implement this on a standard hospital floor where these are not research staff that are implementing these protocols. These are going to be the health professionals on the floor who are part of that clinical setting to implement a research protocol. And the feasibility of that cannot be, you can't assume that that's going to work. Okay, so that's what they decided that they had to do, is see if they could get people who are the regular clinical care to implement these research protocols. Okay. So their usual care group was 70 to 300 milligrams per deciliter. Loose control was 70 to 200, and then the tight control group is the 70 to 100. So you can see where the width is narrowing down. I think they did this in 3 centers. So ultimately what they showed, this is their primary outcome for feasibility. Okay, this is it. So ultimately what they showed is that for people who were in loose control, remember that's kind of, what was it, 70 to 200, that 92% of the people that were randomized, so this was still randomized control trial, 92% were in their range. And then only 44% had the tight control, or within the range that was specified. But what you can see here, so there's their glucose level. Usual care kind of stays at about 160, I guess. Loose control also is coming in at about 160. And you can see with tight control, remember it was supposed to be, what, 100 to 70 to 100? They're much lower than everybody else. So, and this was over the 24-hour period. So from this trial, they were successful in getting the larger trial funded, but it was purely to show that they could implement the intervention and the feasibility of that. The outcome data in terms of, I think it was mortality and neurological function for these people that were critically ill were purely seen as exploratory in the results. There was a little table, and I don't even think the trends were in the right direction, but it was stated as purely exploratory. So ultimately what they said, insulin infusion is feasible and safe. And using this, what the protocol that they had, and that the exploratory efficacy outcomes were at least supportive. But I can tell you the power analysis, I probably should have showed that, but the power analysis for this study was based on the safety, the number of events that they expected, and the differences between the groups in terms of the glucose control. Okay. So there are a lot of words on this slide, but the reason I presented them all is I think it's great. This is probably one of the best examples of a pilot study that was published because it was so much about feasibility. So their objective, and it was a randomized pilot study, they had two different, this is LMWH is low molecular weight heparin, and then UFH is unfractionated, did I get that right, heparin, was to assess the feasibility of conducting a large randomized trial. So they want to do the large trial, but they wanted to see if they could have timely enrollment, can you get people in, get them drugs, so drug administration. Can we get twice weekly lower limb ultrasound? So this is about measurements. So if you're going to stay in a protocol, we have to ultrasound these people twice a week. Can they actually get them ultrasounded? And that's to detect DVTs. Look at bioaccumulation, which would be a safety profile, dose adjustments, and recruitment rates. And so this is all for patients that are in the ICU. So it's very much, the protocol's already set, but it's very much about can they do it, okay? The sample size analysis was based on 120 patients. So this isn't even a small study. 120 people, it's a big study. Probably, I guess in terms of maybe even your site here, this would take a couple of years to recruit this many people. So 120 patients, estimate the proportion of patients who would meet our feasibility criteria using confidence intervals. So this is not based on power analysis, it's based on confidence intervals. And so what they did is they said, well, if 110 out of the 120 patients get their drug the way they're supposed to, then this would be the confidence interval, and the estimate would be 91.7%, and that's the precision that they would have. Okay? And they were willing to accept that. That was tight. That was what they wanted. And then they went further and said, this will be feasible. We're going to say that this study is successful, and we can go on to the next study. Incredibly specific. I think this speaks to the primary author's OCDs. I mean, the fact that they said not just feasible, but we actually have to go in and specify is wonderful. So the first thing they said is 98.5% of the patients have to receive the study drug within 12 hours of randomization. 91.7% of patients receive every scheduled dose. So these are their targets. That many, 91.7%, had their lower limb ultrasounds within 48 hours. Greater than 90% of necessary dose adjustments were made, and then that they can recruit 2 patients per month. So they've got very specific criteria about what it means to be successful to do the next study. And then I think this might be the last one. So this is the BALT study, and the reason that I present this one is it's more of the mechanistic study. So a lot of the studies that I've shown you have been feasibility. Can you implement? Can you do it? Can you recruit? This one is a what I would consider more mechanistic or targeted outcome study, proof of concept that you want to show before you go on to do a trial that has patient-relevant outcomes. Okay. So this, to determine if a sustained infusion of intravenous salbutamol would accelerate the resolution of alveolar edema in adult patients with acute lung injury or ARDS, acute respiratory distress syndrome. So their outcome that they're targeting is basically the resolution of this edema. So they're going to give drug and see if the edema goes down. That's not what we would consider the edema, although a patient may feel that they have edema. It's not what we would consider a patient-relevant outcome like mortality or quality of life or function. So their primary outcome was the extravascular lung water measured by thermodilation at day 7. So this is lung fluid. And they did power the study around this outcome. So for those of us who actually have to power or justify sample size, this would use the typical two-group comparison between the two groups, but the primary outcome now is this lung water. It's not something that's mortality or anything along those lines. So the study was powered to detect a 30% difference in this extravascular lung water between patient on treatment and control groups, 80% power and 0.05. So what they're talking about is a relatively big difference. It's a 30% reduction in this fluid. Okay. And then ultimately, their primary results were based on that outcome. And they're doing, remember I said in the analysis plan, for a pilot, you're probably not going to do p-values and hypothesis testing. It'd be more descriptive and confidence intervals. But for this one, because it's specifically powered around this more proof of concept, they are doing p-values to show that they have a difference. That's what was intended. Okay. Oh, I have one more. Sorry. The last one that I have is the lifestyle interventions and independence for elders pilot study. So I do a lot of work with PTs and exercise scientists looking at physical activity of older adults and different diseases. And this particular study was a pilot study. It was I think I have a sample size later, but it was a multi-center study, a pilot trial. And this was for people who were at risk of disability. So they're 70 to 89 years old. So clinical trials get a lot of heat because we usually don't bring people in that are in this age range. But this is specifically to look at increasing their activity to hopefully to increase their longevity. So they were randomized to a physical activity intervention, and then the control group is a health education group. So in the title is pilot, right? Life P study. So we're going to do the pilot. There's 424 people that were randomized in the pilot. So pilot does not equal small in this case, okay? And so it's multi-center. And so what was going to lead them? What did they need to measure to get them to their big study? So these are all of the objectives for the pilot. Refine key trial design bookmarks, the design, making sure that it works. Sample size calculations for the next study. Looking to see if their recruitment methods are working and refine those. Participant retention. How do you get people into the study but then retain them so that you actually get follow-up and they do the intervention? Then you have adherence and safety of the interventions. Some of it was to set up the organizational infrastructure. How are you going to make sure that people are reporting in things that are going on at the sites? And then lastly, they wanted to see if their outcome for physical activity was valid. And so this short performance battery that's done versus the 400-meter walk speed, those were being done at 6 months and 12 months, and ultimately the larger study would have those as outcomes. So it was pretty clear that they were going to go on, once they had all this information, and they weren't really looking at testing effects so much, but looking at all this information, and then they would move forward. They already had preliminary sample size. Actually, this is ongoing now, I think. It may have just finished. Over 1,600 people are in the LIFE study, the big trial. And their primary outcome is major mobility disability. So for those of you who are either statisticians or work more with outcomes, we now have a categorical outcome. So the pilot trial was mainly focused on a continuous outcome, and now in the larger trial we have a dichotomous one. So it's time to event, major mobility disability. And so here, their primary outcome that
they used to see if at least they were onto something with respect to their intervention was looking to see if they saw differences in the performance battery and also on the 400-meter walk speed. And so they showed that those two outcomes, they do have differences with their intervention groups. So their pilot study is a combination of feasibility, getting everything ready for the larger, but they also tested these kind of, I don't want to say surrogates, but for these continuous measures to see if they saw differences in their groups. Okay. So ultimately they concluded that their intervention was safe, that they changed performance outcomes, which is good, and that their adherence was good as well. Oops. And then they did say that they wanted to go on to look at outcomes that were clinically relevant. So short performance battery and the 400-meter walk speed, those are really more intermediate outcomes, and you want to go on to see if you're actually preventing people from becoming disabled, which is more clinically relevant. So this last one is not a pilot study, it's more of a joke. It's to delineate for you, or to kind of make you a little clearer what the difference is between a pilot and an exploratory study. And I couldn't find anything that sums it up better than this one. So it was a study that was done looking at inhaled and oral corticosteroids. This is for COPD. And they wanted to look at inflammatory biomarkers. So usually when people come to me work with inflammatory biomarkers, they want to look at about 20 of these things. So there were 41 patients, and they were randomized to 3 different arms. They didn't have any power calculations. But the key here that I want you to take home is this is exploratory. This is not a pilot study. And so this really should be more about hypothesis generation, not about feasibility. So this was their table 2, I guess, in their paper. And so let me just give you a little orientation. The biomarkers are on the left-hand side. I think there are about 30. Each one of the treatment arms has 2 columns, one for the effect, the within-group effect, and then its p-value. I don't want you to look at the numbers. There's just too many. And then the p-value to go along with it. Same thing. Each one at treatment arm, p-value, within-group change. And then the last one is prednisone versus its within-group change. I think this one p-value might be a between-group comparison. But ultimately, they let themselves off the hook by saying this is exploratory. We're data dredging. We're hypothesis testing. And so this was their conclusion, which was good. They saved themselves and said some of the cytokines they were looking for were repressed. That was what they were hoping for, was a decrease, and that really a larger clinical trial is needed. So when I think exploratory, lots of outcomes, hypothesis generating, but it is not the last study. There's always more to be done. But this is not what I would call a pilot, and they're not exchangeable. Okay. Regardless of, remember I said that with pilot studies, you still need a good data management plan. And I think a lot of times in the work that I had done with pilot studies, investigators would say, well, I don't have a lot of money, but it doesn't mean that you should shortchange the way that you're collecting your data. So one of the things that, as taxpayers, all of us are paying for the things that NIH is doing. We're all contributing to that. And one of the things that NINDS has done, this is, again, neurological disease and stroke, is they've come up with, it says common data elements, but basically they have a warehouse of case report forms or clinical research forms that you can use so that the data that you collect, even if it's a pilot study, is being collected in a standardized way. And it's as simple as a demographics form, how people are asked about race, ethnicity, income. They have some of the depression forms, some of the scales. Of course, they're very interested in neurological diseases, so they have general CDEs that go across the gamut for all diseases, and that's more demographics and so on. And then they've got some specifically, for example, Parkinson's, Huntington's disease, multiple sclerosis. But I would encourage you, even for any study that you're working on, to not reinvent the wheel, particularly when you're asking things that a lot of studies ask. And then that way you're using what basically they did a lot of focus groups and got consensus from people. This is the best way to collect this data. And so I would suggest that you use these. All you have to do is click on them. They're free. And download them. They're Word documents. and you've got forms that other people are using and have been agreed upon. This is just an example of one of them. This is to collect adverse events for a trial, which is critical in terms of collecting even though it may not be related to the intervention, but collecting things that are happening that the people are experiencing in terms of illnesses or anything along that, anything that's not good. So the adverse event is recorded, the start time, the end time, how severe it was it related to the intervention, what action was taken, and then you have the details about whether it was serious or not, which actually if it's serious it would take you to it, you would need to use another form for a serious adverse event. But just the standard data collection for the adverse events is key, especially if you're working on an intervention study. So this is free, you can download, this is under the general common data elements. Okay, so small studies are important, we're always going to have them, but it's important that your goal and your next steps are clearly delineated. You do need to justify your sample size. I take it quite seriously that every person that I say that you need, that you actually need them. And so we need to make sure that we're not saying that people need to be in studies if they really do not. And you can do some hypothesis testing. It really depends on the type of study. And I think when you go to publish your manuscripts, when you're working with people, or if it's yours, that you need to make sure that you say if it was a pilot, if it really was. So here are all my references. I'll be happy to send out the slides so that you'll have them and read up more on it if you want to design one. And then I'm happy to take questions, although I didn't need much time.
So we have a few minutes for questions.
So what's your top priority when you choose sample size? Because sample size is not based on traditional way. So you think about, you have so many things you want to measure, right? You have feasibility, you want to measure variation. So you have so many things to consider on the budget, how much you want to do. So
Right.
what's your top priority to choose the sample size?
I guess it depends on what the top priority is for the study. I mean, if it really is about feasibility, so how many people does it take to show that you can randomize them, get them in at baseline, randomize them, and follow them up? I mean, if you just want to show that, maybe it only takes 10 people. If you want to be able to get adherence measures, an estimate of adherence, it's going to take more people to measure adherence. And then if you wanted to measure standard deviations, then of course you'd want more. So I guess if they had multiple aims, you maybe would want to take the max across those aims so that you could address all of those. We ran a very informative pilot study in hemophilia, and it was to see if we could recruit people and get them to cross over to another intervention. It ultimately failed. We couldn't recruit 20 people across 5 centers. And what was most informative for that is that the therapies that we were trying to get them to cross over to, people weren't willing to change what they were already on. So even though I set it up as 20 and I had some justification for that, ultimately we failed. But it was very informative why we failed. So I would say if you have lots of aims, I'd probably take the max of whatever that is.
Thank you.
Yeah.
Well, we have time for one more. All right.
Okay.
Well,
Thank you.
join me in